We've now learned about the complex process coupling an action potential in a presynaptic bouton to neurotransmitter release through complex molecular machinery. The action potential firing rate in a neuron makes a large difference about its synaptic transmission. Some neurons, some types of cells in the mammalian brain, fire action potentials at very high rates, almost continuously. So some cells fire at 100 Hz, that's an action potential every 10 milliseconds, almost continuously throughout our lives. Other neurons fire much more sparsely and selectively. They might be silent for minutes and then fire a brief burst of 5 action potentials within a few milliseconds of each other, and then remain silent again for many minutes. There are thus diverse patterns of action potential firing, and the brain has adapted to make use, enhance, and suppress some of the information transmitted by action potentials in different timescales. The amount of neurotransmitter that's released in response to an action potential is therefore dependent upon the history of activity in that particular axon. If an action potential occurs immediately before, then that leaves an imprint upon the synapse, and that can change the amount of neurotransmitter that's released in response to subsequent action potentials. And that process of so-called presynaptic plasticity is what we're going to study in today's video. The modulation of neurotransmitter release in response to activity of that same axon is called presynaptic short-term plasticity, or presynaptic dynamics. It can be thought of as occurring on 3 different timescales, which also relate to 3 different mechanisms that are at play in the presynaptic terminal. We can have short-term synaptic plasticity, that's a form of short-term dynamics that occurs on a timescale of less than 1 second. Typically, 10 milliseconds, 100 milliseconds is the timescale of interaction between neighboring action potentials when we're thinking about short-term synaptic plasticity. It can be divided into 2 forms: facilitation, where subsequent action potentials increase the amount of neurotransmission, and depression, where consecutive action potentials induce less and less release of neurotransmitter. There are other forms of presynaptic plasticity that occur on much longer timescales: post-synaptic potentiation that occurs on the minute timescale, and long-term presynaptic plasticity that can last hours, days, perhaps even a lifetime, and might ultimately contribute to forming memories in the brain. These 2 longer-lasting plasticities rely upon complex signaling cascades downstream of activity and calcium, whereas these are immediately related to the calcium and the amount of neurotransmitter vesicles available. And we'll talk a bit about each of these 4 different forms of presynaptic short-term plasticity in the next slides. We'll begin by thinking about facilitation. In the process of facilitation, we imagine a connected pair of neurons where action potentials in the red neuron propagate down to the presynaptic bouton, calcium influx causes neurotransmitter release, and then postsynaptic potentials are evoked in the blue neuron. One action potential causes calcium influx, causes a postsynaptic potential. Then we envisage a second action potential occurring shortly after the first one, in this case on the timescale of 20 milliseconds later. The second action potential induces a large amount of neurotransmitter release, and therefore also a larger postsynaptic potential. That's the process of facilitation. The mechanism underlying presynaptic short-term facilitation is thought to be an accumulation of calcium in the presynaptic terminal. So the first action potential induces a calcium influx. It activates the voltage-gated calcium channels. Calcium then floods into the presynaptic bouton, and that's what causes neurotransmitter release. There are a number of different calcium binding proteins, of course including the synaptotagmin, that's the effector for neurotransmitter release, but there are many other proteins that also bind calcium. They become calcium buffers. There are also proteins that are involved in sucking the calcium out of the presynaptic terminal, calcium pumps that are involved in reducing the calcium concentration. And so in the 20 milliseconds that follow the action potential, there's a gradual reduction in the calcium concentration in the presynaptic terminal. And that depends on the activity of the pumps, the diffusion of the calcium away from the entry site at the voltage-gated calcium channels, and the binding of various calcium buffers. When the second action potential arrives, there may then be a little bit of residual calcium left in the presynaptic terminal. We may not have reached the equilibrium baseline situation. Equally, some of the buffers may be saturated, and when the second action potential occurs and calcium floods in, those buffers won't be able to bind calcium, and there's already an elevated baseline, and so the peak calcium concentration that's reached in response to the second action potential can be higher than that in response to the first. And even if that's only, say, a 20% increase in the peak calcium concentration, that can make a major difference to the amount of neurotransmitter that's being released. In the last lesson, we discussed that the release rate of vesicles depended upon the fourth power of the concentration of calcium. And so if we have a 20% increase in the calcium concentration, we put that to the fourth power, that induces a doubling in the amount of neurotransmitter release. So even small changes in the amount of peak calcium influx reached in response to an action potential can have a large impact upon the release of neurotransmitter. And in the process of short-term facilitation is thought to be increased calcium signals that give rise to increased amounts of neurotransmitter release. And that's the process of short-term facilitation that typically lasts less than a second and is typically sort of studied on the timescale of action potentials that are 10 milliseconds, 50 milliseconds, or 100 milliseconds apart. There's another process of short-term synaptic plasticity that does the converse, depression. In the process of depression, the second action potential gives rise to a smaller postsynaptic response because of less release of neurotransmitter compared to the first action potential, when those action potentials are separated by a few tens of milliseconds. In the process of synaptic depression, we imagine that the first action potential is quite efficacious, and it releases a large amount of neurotransmitter from the presynaptic nerve terminal. After that neurotransmitter has been released, the vesicles need to replenish. And in the time space of 20 milliseconds, perhaps there's very little time for the vesicles to replenish. And so, in response to the second action potential, less neurotransmitter is released. We can envisage the situation here. In response to the first action potential, there might, for example, be 2 release-ready vesicles. The action potential induces calcium influx. One of these is released and causes a postsynaptic potential. In response to the second action potential, that first vesicle is now gone. It's probably somewhere in the recycling process, trying to load up with neurotransmitter. So there's only one vesicle now that's left, and the action potential causes the same calcium influx, and in this case, there's then a reduced probability of releasing a neurotransmitter. It's also possible to modulate presynaptic release on longer time scales, and this typically involves high-frequency stimulation of the presynaptic nerve terminal and activation of additional signaling pathways. These sort of longer-lasting forms of presynaptic plasticity, they only occur at certain types of synapses. So most types of synapses have the short-term forms of synaptic plasticity, and only certain ones have, for example, post-tetanic potentiation or long-term potentiation that we'll discuss next. And so at synapses that express post-tetanic potentiation, we can imagine having a high-frequency burst of action potential where we might fire the presynaptic cell at 100 Hz for 1 second, so that's 100 action potentials, and each one separated by a 10 millisecond timescale. And we can have a baseline where we stimulate perhaps once every 10 seconds or once every 20 seconds. That gives us our baseline postsynaptic potential. We can plot the amplitude of that in the form of these dots here across time. So we stimulate perhaps every 10 seconds. We see some variation in the postsynaptic potential. Then we give our high-frequency tetanic stimulation, the 100 hertz. That causes a massive influx of calcium through the voltage-gated calcium channels. That rise in calcium activates an enzyme called protein kinase C that depends upon calcium to activate it. So there's a large calcium influx during this 1 second of high-frequency stimulation that activates protein kinase C. And one of the targets of protein kinase C is part of the molecular machinery involved in neurotransmitter release. It's a protein called MUNC18. And if that becomes phosphorylated by protein kinase C, then the release rate at any given calcium concentration is increased. And so if our action potential induces, say, 3 micromolar calcium in the presynaptic cell under baseline conditions, after the post-tetanic potentiation, this 100 hertz stimulus, after the activation of protein kinase C and the phosphorylation of MUNC18, 3 micromolar of calcium will now induce a much higher release rate. And that's then what we see here. After the post-tetanic potentiation stimulation, we go back to our stimulation of action potentials every 10 seconds or so, and we now see that the release of neurotransmitter is enhanced, and it can be enhanced for a time of many seconds. So here we're in seconds, so this is 1 minute, and after 1 minute, there's still a substantial enhancement in the release from this synapse compared to the baseline. So a brief period of high-frequency activity can induce a long-lasting change here over a period of about a minute, where there's a substantial increase in neurotransmitter release. And that can, of course, affect brain function in important and interesting ways, where one can imagine holding a short-term memory of activity at the synapse for a period of a minute. We can also have much longer terms of presynaptic plasticity in so-called long-term potentiation. Again, this only occurs at some types of synapses in the brain. Again, we have a high-frequency train of stimuli in the presynaptic cell that induce the long-term potentiation. We have baseline PSPs that come in, we stimulate every now and then, and we see that there's a relatively reliable postsynaptic potential that comes in. We give our high-frequency tetanization. The amplitude of the PSP that we record postsynaptically increases because of an enhanced release of neurotransmitter. And molecularly, what's thought to be happening at synapses that express long-term potentiation is that the calcium influx during the high-frequency train activates an enzyme called adenylate cyclase. There are calcium-dependent forms of adenylate cyclase, and so the calcium that floods in during the presynaptic train of action potentials activates adenylate cyclase. That in turn turns ATP into cyclic AMP. That's the job of adenylate cyclase. Cyclic AMP is an important signaling molecule that activates another protein called protein kinase A, and that phosphorylates one of our favorite proteins here in the release machinery, a synaptotagmin, not the standard synaptotagmin that's responsible for calcium-dependent release, but another one, another isoform, synaptotagmin 12, at least in some cases where it's been studied. And the phosphorylation of synaptotagmin 12 causes this long-lasting enhancement of neurotransmitter release known as presynaptic long-term potentiation. And this is something that can take place on many minutes, on the hour timescale. So 1 hour, 2 hours later, there's still a large increase in the efficacy of synaptic transmission at the synapse, and that can maybe go on for days. So the amount of neurotransmitter that's being released at a synapse depends on a large number of things. And one of them is the recent history, the activity of that individual bouton in its past. If it just received another action potential before, then the calcium concentration will rise to higher levels with the next action potential, and that can induce presynaptic facilitation, something that occurs on the 10 to 100 millisecond timescale. Equally, the release of a lot of vesicles from the presynaptic terminal leads to a process of depletion. The vesicles need to dock and refill with neurotransmitter before they can be released again. And so there's also a process where we need to refill and reload vesicles. And again, that process of replenishment of the vesicles that are depleted by a previous action potential can take place on the timescale of 10 to 100 milliseconds. So there are these short-term facilitation and depression dynamics that affect nearly all synapses in the brain. Other synapses are able to be modified on longer timescales through signaling processes involving kinases that are calcium sensitive for the most part, and so they can sense the activity of the presynaptic terminal, and through phosphorylating different enzymes or changing the amount and availability of vesicles can then change the efficacy of neurotransmitter release, and some of those modifications can last for minutes or hours. In order to begin thinking about how these modulations of neurotransmitter release can occur in the brain and at different synapses, and in particular when we start thinking about how many neurons interact with each other, it's useful to come up with some computational models, some simple models that nonetheless provide reasonable descriptions about the types of changes that occur in synaptic transmission on different timescales. And so one useful way of thinking about it is by bringing in exponential time courses that will help model different events. And so if we imagine that there's a readily releasable pool of vesicles, and they then need to be replenished, and that will take some sort of time course here that we can give an exponential recovery rate to a readily releasable pool. So we have a given number of resources, and when an action potential is fired, some of those resources are used up. There's an exocytosis, a release of vesicles, and then those vesicles need to recover. And that can be thought of as occurring on some exponential recovery time constant. Equally, we know that there's a process of facilitation, so that when calcium floods into the presynaptic nerve terminal, then the release probability in response to an action potential goes up. And so if we have an action potential, that then causes an increase in release probability, which we can think of as an increase in the calcium concentration, that again lasts for a given amount of time. So we can think of a facilitation time constant. And so we have two different time constants. We can also have, of course, different amplitudes for these effects. And by putting simply two different exponentially dependent time constants together in some sort of releasable pool fashion where action potentials then convert resources into released neurotransmitter, and some of those get used up, we can then generate very complex patterns of interactions we can imagine modeling spike trains with quite complicated dynamics. And it turns out that simple models with just a few variables in them can actually recover quite a lot of the dynamics that occur at real synapses in the brain. And so there are simplifications to the complexity that we see at the molecular level. We can have empirical, much simpler models that do a relatively good job of modeling synaptic transmission, at least on short timescales. And of course, in future work, more detailed models need to be generated in order to fully understand the synaptic process of transmission. And so we've learned that not all action potentials are equal. An action potential will certainly cause calcium influx into the presynaptic bouton and neurotransmitter release, but the amount of neurotransmitter release and the amount of calcium influx depends upon the recent history of action potential firing in that axon. Action potentials occurring within some tens or hundreds of milliseconds of each other can induce facilitation through summation of calcium concentrations in the presynaptic terminal, and they can induce depression through the depletion of synaptic vesicles and a reduction in the readily releasable pool. On longer timescales, the calcium influx that occurs, particularly during high-frequency firing, can induce changes in calcium-activated signaling cascades. Calcium activation of protein kinase C can cause post-tetanic potentiation lasting for a minute or so, whereas calcium activation of adenylate cyclase and cyclic AMP signaling pathways can cause long-lasting forms of potentiation at the presynaptic nerve terminal that probably contributes to the formation of memories. And so changes in the efficacy of presynaptic neurotransmitter release are prominent and form an interesting way in which the brain can auto-regulate its amount of neurotransmitter release from its synaptic terminals.